Hello students, welcome to EPG Pathshala. I am Dr. Vishwanath Tiwari from Central University of Rajasthan. Today I am going to talk about a module that is phase property of biological membrane, principle of membrane organization and its stability. This paper has been taken from membrane biophysics. As you know that membrane is composed of phospholipids in the form of bilayer and a protein is also embedded within it. So, in present module, we will discuss the phase property of biological membrane and principle of membrane organization and its stability. We will also discuss how we can separate the membrane leaflet. As you know, there are two leaflet, outer leaflet and inner leaflet. So, how we can separate the two leaflet and what is the organization of membrane proteins, lipids and their role in the health and diseases. This module has been divided into following section. First we will discuss introduction about the membrane, and then we will discuss how we can separate membrane leaflet. Then we will discuss what are the membrane composition, what is a lipid, what is a protein, what are their organization in the membrane, and then we will discuss how the membrane phase transition takes place and what is the effect of membrane composition on phase transition. Then we will discuss how the protein and lipid are interacting in the membrane and what is the membrane organization, what are the different forces that stabilizes protein DNA, and then we will discuss how the membrane proteins are associated with diseases. And then we have some questions and suggested reading. So, in introduction, the cells are surrounded by biological membrane which mediate several cellular function. They are also playing a role in a specific transportation and protein trafficking. The biological membranes are a highly organized sheet-like structure and is close compartment around the protoplasm and having cytoplasm and extracellular phase. Membrane is diverse in structure and function and share very common features, but they also have dissimilar composition in different organelles. There are mainly consist of two basic components. The first is lipid and second is a protein. And in some cases, the membrane is also having sugar moieties. The membrane is highly selective, permeable to separates their content from surrounding environment and regulating them. All membrane have common general structure in which two lipid layer and having protein embedded within it. The structure is highly fluid and most of the lipid and protein molecule can move across the plane of the membrane. The lipid and protein molecule are held together by non-covalent forces such as hydrophobic forces and van der Waals interaction. Sugar, which is attached to the outer surface of a membrane, are linked by covalent bond with some lipids and known as glycolipids or with the proteins known as glycoprotein. They are found on one side of the membrane, mainly outer side of the plasma membrane and giving membrane asymmetry. As you can see in the membrane structure, we have a phospholipid which is consist of head region and consist of tail region. Head region is present towards the outside or towards the exterior of the cell and tail region is present within the membrane. Proteins which is present in the membrane are two types. Either they are present on one leaflet or they are present throughout the membrane. So, if they are present throughout the membrane, they are known as transmembrane protein. If they are present on one leaflet, they are known as extrinsic protein. Some carbohydrate are also present on one side of the membrane. As you can see that carbohydrate moiety can either attach to the protein, either attach to the lipid. So, they are going to create the membrane asymmetry, and they are the major responsible factor for membrane asymmetry. As you know, the membrane consists of phospholipids and protein. So we will discuss now how we can separate the membrane leaflets, because a membrane have two leaflets, outer leaflet and inner leaflet. So freeze fracture technique is used for the separation of membrane leaflet. In this, a sharp blade fracture a frozen tissue specimen. That fracture line frequently run through the hydrophobic interior of the cell membrane and separating to two phospholipids leaflets. And the integral membrane protein generally remain associated with one of leaflet, either outer leaflet, either inner leaflet. The fractured specimen is then placed in a vacuum and the surface ice is removed by sublimation, a technique known as deep etching or freeze etching technique. After metal shadowing, also known as replica plating, with platinum and carbon, the organic material is removed by acid and leaving a carbon metal replica of the membrane leaflet. And then electron microscopy, especially transmission electron microscopy of the membrane sample, is used to know the membrane proteins as well as the organization of the lipids. In deep freezing studies, cytoplasmic phase of membrane is known as P phase or protoplasmic phase, and exoplasmic phase is known as E phase or extracellular phase, and it is not the mirror image in two phases, and this create pits or holes in one leaflet when it is present in the other leaflet. This may occur because integral protein is bound more tightly to lipid in one leaflet than other leaflet, which leads to the creation of pit where it is loosely bound and it is integrated with the leaflet where it is tightly bound. Now we will discuss the membrane lipids. Cell membrane can form very complex membranous structure as seen in the myelin sheath or in the inner mitochondrial membrane. Different organelles have different lipid composition and is important for determining their shape. Many lipids of the eukaryotic cell are not cylindrical in shape and would not support the formation of membrane bilayer, that is membrane organization or lipid rafts. Three types of lipids found in the biological membrane, which are mainly composed of phospholipids, glycolipids and cholesterol. Phospholipids are the more abundant lipid components of the membrane and it is amphipathic in nature. Amphipathic stands for a molecule which is having hydrophilic properties and hydrophobic properties. So phospholipid have both, that's why they are also amphipathic in nature. Phospholipids are oriented with hydrophobic side chain into the interior of the membrane and hydrophilic head on the exterior surface. This provides the stability to the membrane because exterior of the membrane is polar in nature, so head is going to make interaction with the polar environment, and core of the membrane is hydrophobic, so that hydrophobic side chain stabilizes itself in the core of the membrane, and it is also important for stability and permeability of the membrane. The lipid constitute approximately 50% of the mass of most of the cell membrane, although its proportion varies depending on the type of membrane. For example, plasma membrane have approximately 50% lipid and 50% protein. In all the cell, plasma membrane have several essential functions that includes transporting nutrients into and outside the cell, preventing unwanted material in the extracellular milieu from entering into the cell, preventing loss of needed metabolites and maintaining the ionic composition. It is also important to maintaining the pH of the cell, which is varies from 7.2 to 7.4, osmotic pressure of the cytosol and other functions. To carry out these functions, plasma membrane contain a specific transporter protein that permit the passage of certain small molecule but not other. So you can say they are selectively permeable for a molecule and selectively impermeable for other molecules. Several of these protein uses energy released by ATP hydrolysis to pump ion and other molecule into or out of the cell against the concentration gradient, and this is known as active transport. The smart charged molecules such as ATP and amino acid can diffuse freely within the cytosol but also restricted in their ability to leave or enter it across the plasma membrane. The inner membrane of the mitochondria, on the other hand, contain unusually high fraction of the protein, about 75% of the protein they have, reflecting the abundance of the protein complex mainly involved in the electron transport system, because it is well known that mitochondrial inner membrane is involved in the ATP synthesis, as well as oxidative phosphorylation. So oxidative phosphorylation and electron transport system both are associated with each other, and the protein complexes, 5 complexes which is involved in the ATP synthesis, is present in the as a component of inner mitochondrial membrane. So inner mitochondrial membrane has lipid known as cardiolipin. It is very important that cardiolipin is only found in the mitochondrial inner membrane, and its structure is totally different from phospholipids because it have 4 tails. And cardiolipin, biochemically it is a phosphatidylglycerol phosphoglyceride, and lipid composition of the different cell membrane also varies, similar to the protein variation in the different membranes. For example, plasma membrane of E. coli consists of predominantly phosphatidylethylamine, which constitute 80% of the total lipid, and that means the plasma membrane of E. coli have around 20% of the protein. The mammalian plasma membrane are more complex and containing 4 phospholipids such as phosphatidylcholine, that is also known as lecithin, phosphatidylserine, phosphatidylethanolamine, that is also known as cephalin, and sphingomyelin, which together constitute about 50 to 60% of total membrane lipids. In addition to the phospholipid, the plasma membrane of animal cell also contain glycolipids and cholesterol, which generally corresponds to about 40% of the total lipid molecule. The lecithin is a major phospholipids in animal cell, while cephalin is a predominantly in the bacterial or prokaryotic system. Along with these phospholipid, cholesterol and its derivatives also constitute another important class of membrane lipids, that is known as steroids, and the basic structure of steroid is 4-ring hydrocarbon. Although cholesterol is almost hydrocarbon in composition, it is amphipathic in nature because it have a hydrophilic hydroxyl group and hydrophobic bulky ring. This hydrophilic hydroxyl group interacts with the water, and hydrophobic bulky ring is interacting with the lipid bilayer. Cholesterol is especially abundant in the plasma membrane of mammalian cell, but it is absent in most of the prokaryotic cells, and there are homologs of the cholesterol which is also found in the prokaryotic cells. Around 30 to 50% of the lipid in plant plasma membrane consists of the chloroplast and certain steroids unique in the plants, which is not found in the animal system. Carbohydrates are found in many membranes. As we have discussed that the membrane have protein, membrane have lipids, membrane have cholesterol. Similarly, the carbohydrates are found in the many membrane, and they bound covalently with protein and constitute the glycoprotein, or covalently bound with the lipid and constitute the glycolipids. These bound carbohydrate increases the hydrophilic character of the lipid and protein and helps these protein and lipids to stabilize in the conformation of many membrane protein. That means you can say that the carbohydrate presence makes the survival of existence of protein and lipids in the polar environment. As we know that the outer surface is going to interact mainly with the aqueous environment. So the carbohydrate moiety is mainly dominated in the outer leaflet of the membrane, but they are also found in the inner leaflet. The fraction and percentage is very less in the inner leaflet, but majorly they are dominating in the outer leaflet. The simplest glycolipids, that is glycosyl cerebroside, contain a single glucose unit attached to the ceramide. As we have discussed, there is a membrane lipids. There are another component, that is membrane proteins. So proteins are the major constituent of the cell membrane, and its constitution varies from 25 to 75 on on the basis of various membrane of the cells. The current model of membrane structure is proposed by Singer and Nicholson in 1972 and is views the membrane as a fluid mosaic in which protein is inserted in a lipid bilayer. When phospholipid provides the basic structure organization of the membrane protein carry out the specific function at different membrane of the cells. Without membrane protein, the phospholipid membrane would present in a impenetrable barrier and cell would be unable to communicate with their neighbors. They are unable to perform transport nutrients into cell, or they are going to excrete out waste product outside the cell and response of external stimulus. So, for performing these functions, the membrane protein are important. These protein are divided into two classes based on their nature of association with the membrane. Integral membrane protein are embedded directly within the lipid bilayer, and peripheral membrane protein are not inserted in the lipid bilayer but associated with the membrane indirectly, generally by interacting with integral membrane protein. Membrane protein are the nanomechanics that enable membranes to send or receive messages or transport molecule in and outside the cell and compartmentization. The membrane protein that are present in the particular membrane determine the substance to which it will penetrable and what signal molecule it can recognize. So, membrane protein of the membrane is playing more functional role as compared to the membrane lipid, which providing mainly the building block of the membrane. Now the membrane proteins, many integral membrane protein span in the lipid bilayer with portion of it exposed on both sides of the membrane. It is important for their biological function such as signaling and other functions. The membrane spanning portion of these protein are usually alpha helical region of 20 to 25 non-polar amino acid residues. So you can say that a membrane spanning region or transmembrane region, you can say they are rich in hydrophobic amino acid residue because they have to stabilize itself in the plasma membrane, so they must be hydrophobic. So the hydrophobic side chain of these amino acid interacts with the fatty acid chain of the membrane lipids and they form the alpha helix which neutralizes the polar character of the peptide bond. It is important because the membrane is hydrophobic in nature and peptide bond can create some polarity. So alpha helical structure also important here because they have property to neutralize the peptide bond polar characters. Like phospholipids, the transmembrane protein also form amphipathic structures. This is mainly required in the channels where the hydrophilic portion is exposed towards the channel or towards the side where aqueous environment is there, while hydrophobic portion is towards the membrane side. So in this way it provides amphipathic character to transmembrane protein. So amphipathic character is shown in two ways. In membrane channel protein, if you see one side is totally hydrophobic, which is forming the core of the channel, another side is totally hydrophobic, which interacts with the membrane. But if transmembrane protein is having one side totally hydrophilic and other side totally hydrophobic, then it only form a transmembrane portion where one side is completely bound to the membrane and other side is exposed to the extracellular environment. Some transmembrane protein span the membrane only once, but sometimes it also span multiple times. Most transmembrane proteins of eukaryotic plasmamembrane has been modified by addition of carbohydrate. As I explained, it is present in the form of glycoproteins This is important for cell-cell communication and they are exposed to the surface. The protein can also anchored in the membrane by lipids which covalently attach to the polypeptide chain. Distinct lipid modification anchored protein to cytosolic and extracellular phase of the plasma membrane. Protein can be anchored to the cytosolic phase of the membrane either by addition of 14 carbon fatty acid, that is myristic acid, to their amino terminal, or by addition of 16 carbon fatty acid, that is palmitic acid, or by 15 or 20 carbon fatty acid through cysteine residues. So you can say that 14 and 16 carbon is added to the amino terminal and 15 or 20 prenyl group is added to the sulfur of cysteine side chain. Alternatively, the protein are also anchored to the extracellular phase of the plasma membrane by the addition of glycolipid to their carboxy terminal. These proteins have at least one alpha helical hydrophobic stretch of the amino acid, around 20 residues in the length, which correspond to around 30 angstrom. That means the thickness of an average phospholipid bilayer is around 30 angstrom. If a alpha helical membrane protein span the membrane more than once, it will have more than one of these hydrophobic sections. For example, a calcium ATPase of endoplasmic reticulum and sarcoplasmic reticulum span the membrane 10 times. So it has 10 hydrophobic stretches of around 20 amino acid each. So after discussion of the membrane composition, now we know that membrane have protein, membrane have lipids, membrane also have carbohydrate, and it is going to interact with protein forming glycoprotein or with the lipid forming glycolipids. Now we will discuss the phase transition and membrane fluidity and what is the effect of membrane composition on phase transition. An important property of lipid bilayer is that they behave like two-dimensional fluid. As we know that we have fluid mosaic model where lipids are behaving like fluid and protein is behaving like mosaic into it. So lipid bilayer behave like two-dimensional fluid in which individual molecule are free to rotate and move in the lateral direction. Such fluidity is very crucial for the membrane and it is determined by both the temperature at which the membrane exists and the lipid composition. Because it was found that there is a difference in the membrane fluidity of a thermophilic organism from the mesophilic organism. Similarly, there is a difference in the membrane fluidity from cryophilic organism to mesophilic organism. So you can say the membrane fluidity is mainly influenced by lipid composition and transition between solid crystalline form of membrane to the fluid or liquid form of the membrane is known as phase transition. And this phase transition is influenced by membrane composition such as saturation of the fatty acid, number of carbon in the fatty acid, cholesterol content, as well as the double bond present in the fatty acid, or you can say unsaturated fatty acid. For example, if you see the first case, if we have more saturated fatty acid, it is existing more in the solid phase. Similarly, if you have more carbon in the lipid, it is also existing more in the solid form. That means a membrane with more saturated and more carbon containing lipid enhances more solid phase of the membrane. Similarly, membrane have less saturated or more unsaturated lipids or less carbon containing lipids, it enhances its liquid phase. So the importance of these composition is there when we are talking about the existence of membrane in cryophilic and thermophilic conditions. A thermophile have more saturated and more carbon containing lipids, while a cryophile have more unsaturated and less carbon containing lipids. Each addition of, as we say that we have carbon in the lipids which influence its solid nature, so each addition of one CH2 group make a favorable contribution about 0.5 kilocalorie per mole of the free energy, and because free energy must have negative value for a favorable free energy, so minus 0.5 kilocalorie per mole free energy interaction of adjacent hydrophobic chain. So if you increase the more and more CH2 group, more and more free energy for its interaction increases, which is favorable for stability of the membrane. Similarly, cholesterol, as I told that it is having a hydrophobic group, which is hydrophilic hydroxyl group, and hydrophobic bulky side chain, which is flexible, and hydrophilic hydroxyl group interacts with the phospholipid head, while hydrocarbon or bulky hydrocarbon tail interacts with the non-polar core of the bilayer. So in this way, this cholesterol, by interacting to the phospholipid head by its hydroxyl group and tail of the phospholipid by its hydrocarbon bulky tail, make the existence of cholesterol stable in the membrane. As the temperature below the Tm, Tm is a temperature at which a molecule exists in 50% solid and 50% liquid form. As we have discussed that cholesterol have interaction with the phospholipid by its hydroxyl side chain hydroxyl group with the polar head of the phospholipid and bulky hydrocarbon tail with the tail of the phospholipid. So how the cholesterol influence the fluidity of the membrane, it is very important, and the behavior of the cholesterol is very biphasic. So at the temperature below the Tm, the cholesterol interferes with the interaction of the hydrocarbon tail of the phospholipid. Therefore, it increases the fluidity. But at the temperature above the Tm, cholesterol limits disorder because it is more rigid than hydrocarbon tail of the fatty acid and cannot move in the membrane to some extent. Therefore, it limits the fluidity. That means if you see the cholesterol, if you put cholesterol at the temperature below the Tm, it increases the fluidity. But when you put the cholesterol above its the Tm, then it decreases the fluidity or it limits the fluidity. Metal ions such as calcium decreases the fluidity of the membrane because of interaction with negative charge phospholipids, which reduces the repulsion between polar group and increasing the packaging of lipid molecule. It is noted that E. coli, the ratio of saturated to unsaturated fatty acid decreases from 1.6 to 1 when the growth temperature shifted from 42 degree to 27 degree centigrade. That means if you decrease the temperature, more unsaturated fatty acid will express. It is also noted that the ratio of saturated to unsaturated fatty acid lower in the winter than summer in some species of the wheat. This is again a adaptation of the wheat to encounter the temperature. This phase transition leads to the membrane fluidity, which increases the permeability of the water and other hydrophobic molecules and lateral movement of the integral proteins. The free energy barrier of flip-flop of the protein molecule is even larger than the lipid because extensive polar region the protein have. Hence, the flip-flop the protein molecule is not generally observed, but it is mainly observed in the case of lipids. After the phase transition, let's have discussed how the protein and lipids interacts and how they are going to influence the membrane stability and organization. So as we know, the cell membrane is crowded with the protein that have different affinity with the lipid raft or raft domain. So what makes the transmembrane protein raftophilic? Although the protein is hydrophilic in nature and lipid raft are hydrophobic in nature. To study it, so Fersen tag transmembrane protein expressed in the cell and their partitioning in the phase separated plasma membrane can be measured by quantitatively using confocal microscopy. And Leventhal et al. Showed that palmitylation play an important role in regulating raft affinity for these proteins. The reason for the exclusion of the raft protein from raft phase in the chemically induced bulbs was the use of dithioerythritol, which leads to the depalmitization by thioester reduction. So in this way, palmitylation has been created on the transmembrane protein and it is removed by dithioerythritol. because cysteine palmitization is only post-translational lipid modification of the protein that has been shown in reversible regulation manner. So these data suggest that role of palmitylation as dynamic RAF targeting mechanism for transmembrane protein. However, it is important to point out that palmitylation is definitely not sufficient for RAF association. There are some other mechanism because there are many palmitylated protein which is not RAFT associated, including non-RAFT marker like transferrin receptors. So it was concluded that palmitylation is one of the mechanism which is making transmembrane protein rafidophilic, but it is not always that palmitylation leads to the rafophilic transmembrane protein as seen in the transferrin receptor. Both TMD length, transmembrane domain length and amino acid sequence will be involved in the defining rapidophilic nature of the transmembrane protein. Hydrophobic interaction is a major stabilizing force between transmembrane domain of the membrane protein and phospholipid. As we know that membrane is hydrophobic in nature, so hydrophobic interaction along with the van der Waals forces are the forces which stabilizes these membrane into membrane lipid complex, and they are important for lipid protein interaction and the stability and organization of the membrane. So hydrophobic interaction are non-covalent interaction that minimizes the contact of the hydrophobic or non-polar molecule with the aqueous environment or amphipathic molecules. That means it provided tendency of non-polar molecule to aggregate in the solution, and this is the reason why membrane is a organized structure where the hydrophobic fraction of the protein and hydrophobic part of the lipids are clustered together or aggregated together in a supramolecular membrane structure or membrane organized structure. And the hydrophobic amino acid such as isoleucine, valine, leucine, phenylalanine, methionine and cysteine of the transmembrane proteins are playing significant role in the membrane protein interaction or lipid protein interaction. Fatty acids of the phospholipids and other lipids interacts with these amino acids because fatty acid is hydrophobic in nature. So protein when form channel in the membrane, they must be amphipathic in nature, which further enhances the stability of the channel in the membrane and allows molecule to pass through it. And the energy of hydrophobic interaction formed by methylene group varies from 0.5 to 2 kilocalorie per mole, while the energy of hydrophobic interaction contributed by isoleucine, valine, leucine can have energy up to 5 kilocalorie per mole. After discussion with the interaction of membrane with the protein and lipids, so membrane protein and membrane lipid interacts with the hydrophobic and hydrophilic interaction, but they are mainly dominated by hydrophobic interaction and van der Waals forces. Similarly, we have discussed how the membrane composition influencing the fluidity of the membrane. So we will discuss how our knowledge on the membrane protein helps new drug designing to treat the diseases. As we know that there is a membrane is playing very significant role in different functions and their defect leads to different diseases. For example, if we have non-functional ion channel, then there is a disease known as cystic fibrosis. So cystic fibrosis is an autosomal recessive disease that results from mutation in CFTR, which is a chloride channel. Not only that, membrane protein also act as a entry point for the viruses. For example, GP20, GP120 of HIV, the surface protein, interacts with the CD4 protein on the surface of T cells. So GP120 of HIV interact with the CD4 of T cells. Similarly, various toxins interfere with the transmission of the messages across biological membrane. For example, tetanus neurotoxin and botulinum neurotoxin are protein toxin that affect nerve impulse transmission between nerves and muscles. Tetanus toxin binds glycolipid enriched at presynaptic membrane at motor neurons and exert this function. So here you have also seen how the glycolipid interaction with the toxin is leads to the pathogenic condition. Not only that, these proteins, membrane proteins, also target of the drugs. An important class of the drug target is pore-forming membrane protein encoded by the viruses. HIV, influenza and polio among other viruses includes membrane protein that form pore in the host cell. For example, pore-forming BAPU protein from HIV-1. So in this way we can see that membrane protein and lipids also play important role in creating different diseases, or you can say viruses uses these protein for their infection. So when we manipulate the membrane protein and membrane lipids, or we will monitor the interaction between pathogens and membrane protein and lipid, it will help us to design drug or cure some diseases. So let's summarize what we have learned in this module. So in present module, we have discussed what are the membrane composition. We have discussed the membrane lipids, we have discussed the membrane protein, and we have also discussed the phase transition of the membrane, and we have discussed the phase transition of the membrane will be altered by composition of the membrane. So we can say the phase transition will alter the membrane fluidity, which finally alter the function of the membrane. And we have discussed that how a thermophilic membrane is differ from a cryophilic membrane. We have also discussed the different protein and lipid found in the membrane and their interaction. We have also explained the interaction of protein in the lipid raft found in the membrane. We have discussed the role of different components of the membrane in phase transition, and we have also discussed what is the role of saturated, unsaturated fatty acid, what is the role of cholesterol, and what is the role of metal ion in phase transition of the membrane.